So, uh, hello to everyone, and I welcome you in this video. In this video, we'll be um, discussing a question that is on carbohydrates and sugars. And this question, which I have taken from 2014 biochemistry question paper, and this is question number 5. Here the question states as choose the option with 2 reducing sugar. So here we have there are 2 sugars in a group given in 4 options, and we have to tell which one is the reducing sugar. So okay, so very firstly let's talk about the sugars. So we have sugars, single unit of the sugar, which is called as uh, monosaccharide, monosaccharides, and disaccharides, where two single monosaccharides makes a bond and make the disaccharide sugar. Okay, the disaccharide one. So here, if I talk, so here we have, firstly we can talk about the very basic sugar, that is the glucose. So if I'll make the uh, structure of acyclic structure of glucose, so these sugar are divided into two parts. One is the this one is this aldose sugar, and another is the ketose sugar. So in aldose we have the functional group aldehyde. However, in in ketose we have the functional group of ketone. So they are generally a 6 carbon ring or structure. So, and this is the 6 carbon chain. So glucose, we can write it is a aldose sugar, so it is an aldehyde. This is the aldehyde group, and uh, bottom we have ending with CH2OH. Okay, and in between we have OH and H are arranged in groups. So okay, so here we have, this is 6 carbon structure, and this is the structure. So here if we see, this is the this particular OH group is in symmetry with the last carbon. So both the OH are in the same side, that is why it is D-glucose structure. Okay, this is the D-glucose, and here we have to see the last carbon orientation. If the OH is in the same orientation of the last carbon or OH, then it is the D-glucose. Otherwise, if the OH is on the opposite side, then it will become L-glucose. So the last carbon just before the group, the last functional group, if the OH group is in the same side, it will be the D-glucose. Otherwise, the opposite side will come the L-glucose. So we have the D-glucose structure with us. So this is the D-glucose. Okay, so that is D and L are the two forms of glucose. Other than that, this same structure, uh, because of the rearrangement of these hydroxy groups, they form different epimers. So if I write the epimers, so glucose have so here this is the first carbon where the aldehyde group is there, second carbon, third carbon, fourth carbon, fifth carbon, and the sixth carbon. Okay, so we, if I'll make another, so here the aldehyde group. Okay, so another epimer. So here in the second carbon you can see OH group is present this side, but in this epimer this OH is present opposite side, and rest of the arrangements remains the same. So this is another this is, we can say it as C2 epimer, C2 epimer, and this is the structure of mannose, and this also, because of this, it is called as D mannose, D mannose. Okay, other than this, if we leave this one as it is at second position, and at the fourth position, if so 1 2 3 4 fourth position, if we have the organization like this, so then this become the C4 epimer, and this is called the galactose. Okay, so galactose is also a epimer of glucose. Okay, so this is how these rearrangements make the different epimers, even though the structure and the composition is the same. Okay, so these are the different epimers. And other than that, we have ketose sugar. So if I write a ketose sugar, so compared to um, glucose, so we have the ketose sugar arranged like this. This is the ketose. This is the ketose. Okay, so this is the first carbon, the second, third, fourth, fifth, and sixth carbon. Okay, so this is also D-fructose. Here, the functional group is this keto group. Okay. However, in glucose, we have the functional aldehyde. This is my glucose, and this is the fructose. So this is the acyclic form of these sugars. So for the cyclic form, what happens here, for the so when in between this particular linkage, if they form a bond between the aldehyde, if they form a bond between the aldehyde group and the alcohol group, it is called as the hemiacetal bond formation. Okay, and because of the hemiacetal bond formation, they make the cyclic form. So what happens here is, so here what happens, so this particular one, so becomes like this, and this is the hemiacetal bond that is being formed, and this can be converted in the cyclic form. So in the cyclic form, okay, so here comes this particular oxygen, and these are the arrangements of the sugars. So their arrangement, what we can see is the first carbon where aldehyde group was present. Okay. Carbon. So we have 2, 3, 4, 5, and 6 carbon. This is the 6 carbon, uh, 6 carbon structure or cyclic structure of the glucose. Okay. So this is where the functional group resides, which can be freed. Okay. So we can write it as, so there is an equilibrium between the open, the cyclic ring structure and the acyclic ring structure, so that equilibrium is maintained. So this aldehyde group can be functional. So this aldehyde group can be functional, and this aldehyde can further, okay, this aldehyde can be reactive, and this reactive aldehyde group can undergo oxidation and make the acid, respective acid form of this sugar, from aldehyde to acid. Similarly, in the fructose 1, so in the fructose 1, what happens? There is a bond formation between this and this. So here in this particular orientation, this OH and H, if OH is on the top, it is the beta configuration. This is the beta uh, uh, D glucose, you can write, and if this OH is down, it is the alpha glucose. Okay, so this is the beta D glucose, and if the OH is down, this OH is in opposite orientation, it will be the alpha D glucose. And if in the fructose structure, what is happening here? In the fructose structure also, so here it becomes an OH, and then, okay, so that is what happens, and this cyclic representation is shown. It become a furanose ring, and in case of glucose, that becomes a pyranose ring. Okay. This is 2OH, this is here 2OH. Okay, so this is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon. This is the furanose ring. So this is also fructose, cyclic fructose form. And here the second carbon, which was the ketone form, so here also there is an equilibrium. So this one is hemiacetal, this one is hemiketal. So there also an equilibrium between the cyclic and the acyclic state forms of the sugar. So because of the presence of the active keto and active aldehyde group in their structure, these sugar work as a reducing agents, and then that's why they are called as reducing sugars, because they get oxidized into the respective acid form and reduces the divalent cations for a or divalent cations or other ions like cupric ion and ferric ion. They oxidize those ions and reduce those ions and undergo oxidation. So that is why they um, serve as a reducing sugars. However, in case of disaccharide, if I will draw a structure for you. So in case of, suppose disaccharide, so we have a structure. Suppose I'll be writing maltose. So maltose is a dimer of glucose. So alpha D glucose and alpha D glucose, it is a dimer of alpha D glucose. So here So in both are of alpha D glucose. For alpha, we have the OH group in the down. So here the condensation happens, release of water takes place. So here this is the first carbon, second carbon, third carbon, fourth, fifth. Okay, this is the first, second, third, fourth, fifth, and sixth. So there is a linkage forms between these two. So water being removed, and then there is a, so this is the O glycosidic linkage, or you can say alpha 1 4 alpha 1 4 glycosidic linkage. So here, in case of this particular alpha D glucose, the first carbon or the anomeric carbon where the reactive aldehyde group is present, now it is in locked conformation, and that group is not free because of the disaccharide bond formation. But this one have the first carbon where the anomeric carbon is the aldehyde group is free. That is why this particular sugar, that is maltose, it works as a reducing sugar. So that is why it is a reducing sugar. However, if I'll see the structure of sucrose, so in case of sucrose, what happens, sucrose is also a disaccharide. So in sucrose is a combination of glucose and fructose. So here, in case of glucose in case of sucrose, we have beta, or we have beta or alpha 1 and 2 beta. So this glucose is in alpha configuration, alpha glucose, alpha D glucose, and fructose is the beta fructose which is being used. So that is why this particular type of bond formation takes place. Okay, so that is why this particular here, this is the second carbon, this is the first carbon. So when these 1 2 glycosidic bond gets formed, alpha 1 beta uh, 2, this linkage is being formed. So here, from the glucose, the first carbon that harbors the aldehyde is again involved in the involved in the bond formation and glycosidic bond formation, and the second carbon, which is the keto active group of this fructose, that is also involved in the bond formation. That is why no functional group is left to get oxidized and work as a reducing sugar. That is why this particular one, sucrose, is not a reducing sugar. Okay, so let's go and see our options. So we have these four options, four combination. So here, in case of lactose and maltose, so lactose is a combination of and galactose and glucose. Lactose is a combination of galactose and glucose, and in galactose and glucose also we have 1,4 glycosidic linkage. So that is why one end of glucose is free for the aldehyde group. In maltose also we saw maltose is glucose, so that is also 1,4 glycosidic linkage. That is why the aldehyde group carbon, the anomeric carbon, is free, the first carbon and one of the monosaccharide. So this here both the sugars work as a reducing sugar. Let's see if option C B. So in the second option there is a trehalose and sucrose. So sucrose we just now discussed, 1,2 glycosidic linkage. In fructose and glucose there also it is both are not free, both functional groups are not free. However, in the trehalose, that is also glucose and glucose dimer, but the linkage is one glycosidic linkage. So that is why in trehalose it is, there is no aldehyde group is free. Similarly, in the maltose is reducing sugar, but trehalose becomes non-reducing because of the no functional group available. And here also only lactose is the reducing sugar and sucrose is a non-reducing sugar. So out of the four pairs, the first pair, which is the lactose and maltose, is the reducing sugar pair. Okay, so the correct option will be the option A for the two reducing sugars. So this is the answer of the question, and this is the NPTEL reference, and here you can study more about the detailed structure of these monosaccharide disaccharides, their different forms and their reducing properties. Thank you.